In the last lecture, we have discussed about the polysaccharides, which are having an important role in the energy storage form. In this video, we will discuss about the structural components of polysaccharides. The first component is cellulose. Okay, so cellulose is a homopolymer of D-glucose and it is linked by beta 1,4 linkage. It is present in the cell wall of plants from algae to angiosperms. It is also present in the cell wall of oomycetes. Oomycetes are a kind of fungi. Okay, oomycetes is a category of fungi. So it is present in the cell wall of oomycetes, not all the fungi. Okay, its structure is beta pleated sheets. So if it is sheet-like structure, then the bond is interchain hydrogen bond. Okay, this cellulose is synthesized by a plasma membrane enzyme called as cellulase या cellulose synthase. This enzyme is present in our plasma membrane or the plasma membrane which synthesize the cellulose. Okay, this cellulose is not digested in our body because we lack the enzyme cellulase. This enzyme cellulase digests the cellulose, but as we lack it, we cannot digest the component of cellulose. So, whatever plant material we take in our diet, we take it as a roughage. All the cellulose is taken as a roughage in our body. Okay, ruminants, if we talk about the ruminants, they have special chamber in their stomach that is rumen. Okay, in that rumen, they harvest the microbes, and what these microbes do, they convert this cellulose into glucose and then into fatty acids, short chain fatty acids, and they digest this cellulose. Okay, the next the next component is of the next structural component which we have to study is chitin. Okay, so chitin is a homopolymer of N-acetyl D-glucosamine. It is a homopolymer of N-acetyl D-glucosamine. Okay, with linkage of beta 1,4 linkage. Okay, it is also having a sheet-like structure like the cellulose. And if it is a sheet-like structure, then the bonding is interchain hydrogen bonds. Okay, this chitin is present in the exoskeleton of insects and cell wall of fungi. All the fungi except oomycetes, because in oomycetes the cell wall is composed of cellulose, and it is the second most abundant biomolecule. The first most abundant biomolecule was cellulose. Okay, it is the this cellulose is the most abundant biomolecule. Okay, next component which we have to study in the structural component is pectin. Okay, so pectin is a homopolymer of galacto-uronic acid, and the linkage is alpha 1,4 linkage. Okay, it is a component of middle lamella. That means कि it acts as a cementing agent between two cells. Okay, it is also having a sheet-like structure like other structural polysaccharides, which is having an interchain hydrogen bonds. Okay, so this was all about the homopolysaccharides. Now we will discuss some of the heteropolysaccharides. Okay, heteropolysaccharides. As I have told you earlier, that the heteropolysaccharides are made up of different sugar subunits. Okay, different sugar subunits. If but generally, if there are two sugar subunits, that generally happens. So the two sugar will be the first sugar and the second sugar. The first sugar must be a acidic sugar, okay, and the second sugar must be a amino sugar, okay. So what are the examples of acidic sugar? Like glucouronic acid, galactouronic acid, muramic acid, idouronic acid. If it is an amino sugar, then it can be N-acetyl D, glucosamine, or N-acetyl D, galactosamine. Okay, these heteropolysaccharides are generally charged molecules. They are having a charged, they are basically charged molecules. Okay, and the linkage they have, in general senses, the linkage they have is beta 1,4 glycosidic linkage, or it can be also beta 1,3, but most preferred linkage is beta 1,4 linkage. Okay, one example of a heteropolysaccharide is a bacterial cell wall component, that is peptidoglycan. Peptidoglycan is present in the bacterial cell wall. Okay, this glycan moiety of the peptidoglycan is composed of two sugar molecules. The acidic sugar is N-acetyl muramic acid, and the amino sugar is N-acetyl D glucosamine. Okay, and the linkage is beta 1,4 linkage. So in our secretions, in our secretions of our body, like tears, saliva, we have an enzyme called lysozyme. Okay, this lysozyme attacks on the beta 1,4 linkage and breaks the cell wall of bacteria. Okay, some other heteropolysaccharides are present in our ECM. What is ECM? ECM is our extracellular matrix. Okay, we know that in our body, all the cells of our body are held together in a space that is the extracellular matrix. Suppose this is the extracellular matrix. So this substance is collagen. Okay, and this liquid material which is filled over here. Okay, this is what is having a component, that is glycan, or most usually we can say a heteropolysaccharide. Okay, and a heteropolysaccharide, like what kind of heteropolysaccharide can be present in the extracellular matrix are hyaluronic acid, okay, and dermatan sulfate, keratin sulfate, okay, chondroitin sulfate. These all are peptidoglycans which are present in our extracellular matrix. Okay, this is not a peptidoglycan. Okay, so this was all about the polysaccharides. homopolysaccharides and heteropolysaccharides. If you like this video, hit the like button and subscribe my YouTube channel. Thanks for watching.